Hi there, my name's Kim Komatsu, and I've been tasked with talking to you today about how communities respond to environmental conditions and thinking about species manipulations in an experimental context. I'll start out by talking about a case study example that I think really exemplifies why thinking about community composition matters. So this case study example comes from a tallgrass prairie ecosystem, the Kanza Prairie Long-Term Ecological Research Station, where over a period of about 20 years, experimenters were doing water additions in this prairie system, so irrigating our plots. And we can see over the first 8 years of the experiment, tracking above ground net primary productivity, so the growth of the plants in the system, as these gray bars. And then we were able to predict or model that productivity using a process-based ecosystem model. And that's shown here in the green dots. And so for the first 8 years of our experiment, we see that our model is really well able to predict the above ground net primary productivity values in this experimental system. But after 8 years, we saw a big switch in the dominant species in these plots. So Andropogon gerardii was traditionally the dominant grass species in this system, and in these irrigated plots, that species declined very greatly in abundance. And it was replaced by Panicum virgatum, which is another C4 grass. So here we don't have a change in the functional group that dominates these plots, but we do have a change in the identity of the dominant species. And after this community change, we see that our process-based ecosystem model is really poorly able to predict productivity in the majority of years in these plots. And so thinking about that switch in the identity of the dominant species or the community composition, in this case, really matters for our ability to predict the functioning of this system with increasing water availability. And so thinking about community composition, we know that it's important, and we could think about lots of different ways that we could measure community composition. And so I'm just going to run down a few different metrics that we could use to think about how communities are organized. First is a really common metric, something that we think about a lot as ecologists, which is just the number of species in an ecosystem, or plant species richness. And so we can look at how plant species richness is changing around the world, both observationally and in our experimental contexts. Just thinking observationally, this figure is showing through time on the x-axis a bunch of different time series data looking at the log of species richness on the y-axis. So again, this is just the number of species that are found in individual samples through time. And each individual line here is showing that an individual time series in a variety of different ecosystem types around the world. And so we see in some cases species richness is declining over time. In other cases, species richness is increasing over time. And this black line down the middle is showing our common global trend. And so we're seeing here that across all of these ecosystems through time around the world, we don't see a response of species richness. So species richness looks to be constant globally through time. We can also think about an experimental context. So here we're looking at a variety of different global change experiments around the world where people have manipulated things like water availability or CO2 availability in the atmosphere or nitrogen availability in the soil, temperature, etc. And across the x-axis, we're looking at the number of years that global change treatment has been chronically imposed. And the y-axis is showing our proportional difference in richness between our treated plots and our control plots. So here, a value of 1 would mean we see a 100% increase in species richness. A value of -1 would mean we see a loss of all of our species in the system. And again, each individual line shows a different global change treatment within an experiment from somewhere around the globe. And this thick black line down the middle is showing our global trend. And here again, we're seeing globally a trend that there is not a change in species richness with this wide variety of global change drivers that we are experimentally imposing. And so we can say here that it looks like species richness is not a very sensitive metric of community responses in our ecosystems around the world, either looking at these observational or experimental trends. And so then what is this missing? What is plant species richness as a metric missing? And I would argue that it's missing thinking about the identities of those species as well as potentially their abundances. And so we could look at multivariate metrics of community similarity or dissimilarity to try and get at these aspects. So here looking at those same data sets, observational data sets that we use to look at richness, now we can look at those community similarity. And so here we're looking at how similar our communities are thinking about species identities, where a value of 0 means that there's no shared species, and a value of 1 means there's complete overlap in the species present in our communities through time. And so looking across time on the x-axis, we're seeing this global trend that communities become more and more dissimilar from each other through time in our time series data sets. And we can also look at our experimental framework. So here we used a different metric of community, multivariate community change. Now we're looking at community dissimilarity. And so here this metric takes into account not only the species identities, as the similarity metric did, but we're also looking at species abundances. And here a value of 0 will mean our communities are completely dissimilar, there's no shared species, and a value of 1 would mean that all of our species are exactly the same, and not only are they the same species present, but they're also present in the exact same abundances in one plot versus another. And so here again, looking through time on the x-axis in our experiments, we see this global trend. Our thick black line is showing that communities are becoming more and more dissimilar from each other in treated plots where a global change treatment is imposed compared to our control plots. And so here we're seeing that communities are changing globally, and these multivariate metrics are able to better assess what those community changes are. But I would say that these multivariate metrics of community change, similarity or dissimilarity, are still relatively opaque. So we have a hard time saying what exactly is changing about these communities. And so we could start to break down what our various metrics are that we could think about our community composition to try and get at more nuanced aspects of the community. And so to illustrate this, we could think about rank abundance curves. So these are arraying our species in our plant community along the x-axis in order of their rank, from the most abundant species all the way down through the least most abundant species. And then we're plotting them against their abundance on the y-axis. And so we might imagine that we have a community that we might sample at a certain time period, or it might be our control plot, and we have a dominant species, maybe some subordinate and some rare species in our community. And this is just an example of a photo of some plots that I work in a grassland system, where we see a dominant grass species as well as a few subordinate forb species or rare forb species in the system. And we might compare that to another community either at a different time point or in our global change treatments, and we could see a variety of different ways these communities could change. We could think about changes in evenness. So here, what used to be the most dominant species, that purple species, has decreased in abundance, and all of our species have more similar abundances to each other than they did in our initial community. And so this photo illustrates now we see a decline in the abundance of those dominant grasses and a more even community with these flowering forb species. We might also see a reorder or a rank change. So here we have what used to be our most rare species in the community coming to dominate, and what used to be the most dominant species dropping down to be relatively rare. And so we again have all the same species in our community, they're just changing their relative abundances relative to each other. And again, this is illustrated here by this grass species is now very rare within this community, and these forb species that were initially present in low abundances have now come to dominate the system. In addition to changes in the existing species within the community, we of course could also see losses of species. So here that blue species has disappeared, or we could see gains of species. So here a red species has appeared. And this is distinct from thinking about just plant species richness, because if we had a loss of a blue species and a gain of a red species, there would be no change in the number of species in our communities, but we do have two species different in those new communities that are establishing. And so here when we're looking across a variety of these global change experiments, again, where we're imposing global change drivers such as nutrient availability or increasing atmospheric CO2 or irrigation or drought experiments, we see that evenness change occurs in these experiments about 30% of the time, as does rank change, species gains, and species losses. And overall, about 71% of our communities experience at least one of these aspects of community change across more than 450 different experimental treatments that we were looking at, or experimental communities we were looking at. And many of these communities actually experience multiple different aspects of these community changes all at once. And what's, again, really important about this is that these community responses that we're seeing are linking to functional responses. And so here, looking at that community dissimilarity metric across the x-axis, again, this is that multivariate metric of community change that takes into account both species identities and their abundances, where higher values are indicating communities are more different from control plots. And we can link that to our productivity differences. So above ground net primary productivity in this system between treatment and control plots, where again, higher values are indicating a greater difference in productivity between treatment and controls. And what we're seeing is that community dissimilarity directly links on to our productivity difference that we're seeing across these global change treatments. And when we break it down into our various different aspects of global change, we again see strong relationships with these community change metrics like rank change and our productivity responses in these experiments. And so rank change, again, is thinking about going back to our case study example where we're seeing one dominant species decline in abundance and being replaced by another species that was already present within the community. Also think about the evenness change, species gains and species losses. I'm not going to show you the figures here today, but when we add up all of these different aspects of community change into our model, our total predictive power of our statistical model is about 50% of the variance in the productivity difference is being explained by these community changes that we're seeing, these very specific community changes. And this is really remarkable because we know that if you add fertilizer to plots, if you add water to plots, or if you remove water, if you drought your plots, you're going to see physiological responses of these plants. And so the fact that 50% of the variance in our productivity difference in these global change experiments is being driven by these community changes is really remarkable. And I think it really touches on why it's so important to think about these community changes when we're thinking globally and trying to predict how communities or how ecosystem function is going to respond to global change going forward. So if we're thinking about shifts on our environmental drivers, of course we know that organismal traits can be changing. They can be physiologically responding to these global change shifts. But we also know that community structure is really important when we're trying to predict ecosystem function. And of course, all of these different factors interact with each other as well. And so we want to come up with a more mechanistic understanding of how communities are changing and what that means for the functioning of our systems. And so a great way to try and get at these mechanistic understandings is about experimentally manipulating our communities. And so there's two main ways that people have thought about experimentally and successfully experimentally manipulated plant communities. The first is thinking about communities that are artificially assembled. So there's a wide variety of biodiversity ecosystem function studies all around the world now. They're very common in grassland systems, which is where these ideas kind of originated. But there's many new forest experiments as well, where people are planting different numbers of species or different types of species within these monoculture and diverse plots of trees as well as grasses in these systems. And so what have we learned from these experiments? So this figure is looking across a variety of different ecosystem types on the x-axis at the log response ratio of net primary productivity, thinking about diverse communities compared to monocultures. So here a value of 1 is indicating that diverse communities have increased productivity compared to monocultures, while a value of -1 would show decreased productivity. And we see across a lot of these different ecosystem types that there is an effective biodiversity treatment driving ecosystem function. And it's not shown here, but we also see a lot of evidence that biodiversity may be driving the stability of ecosystem function as well. But what's really interesting is that we do have limited data from a variety of different ecosystem types, including temperate forests. And we see that these other ecosystem types don't necessarily show the same trend, or certainly not as strong of a trend. And so specifically thinking about our temperate forests, they sit right here, the mean response is right here at 0 with variance around that mean. And so having more and more experiments that are directly manipulating diversity in experimental context in temperate forests is really key here. And this is something that is occurring currently. There's now, I think, more than 30 different experiments around the world that have manipulated tree diversity within temperate forests but that number certainly could increase as well. So thinking about assembled communities, these BEF experiments, what are some limitations? So one common criticism of these experiments is that they might not have direct applicability to nature. So many of these experiments are imposing random species loss. So we see random combinations of species that are being assembled or planted in these communities. And we know that with global change, loss of biodiversity around the world, we're not necessarily seeing random species loss, and many don't account for non-random species loss, although some are starting to do this now. These experiments have a really high initial setup effort, so it takes a lot of effort, especially in the forested systems, to plant all of these species. And then they also take a lot of effort to maintain. And that maintenance of these biodiversity treatments really negates natural processes of dispersal of new species into these plots, or changes in species composition that are of species that are not supposed to be present in those experimental treatments. And then finally, of course, these assembled communities are also really difficult to scale. So it's hard to say what would be happening at a larger spatial scale than the plots that we're able to maintain. So another really common way that we experimentally manipulate communities is by removing species. So this is just an example from, again, the tallgrass prairie system, where the dominant species has been removed and only the rare grass species and the forb species remain in this community. And so looking across a variety of ecosystem types, including not only grasslands, but also forests and other ecosystems, we can look at the log response of diversity metrics. So thinking about things like richness, Shannon's diversity, evenness in a meta-analysis framework. And we see a trend towards increasing diversity when you remove the dominant species from these systems. And that makes sense because these more rare species are being released from competitive effects of the dominant. But what we see, even despite that increase in richness or increase in diversity in our communities, we see a decline in function. And these are functions including not only net primary productivity, but also things like nutrient cycling or carbon storage in the soil in these ecosystems. And so across our ecosystem types, we're saying that dominant species removals really looks like it's driving functional decline. And now thinking specifically about a tallgrass prairie system, we were able to compare the effects of removing dominant species, which is this large green bar here, which we know had a negative effect on productivity in the system, to a wide variety of other global change drivers. And we see that removing those dominant species had almost an equal and opposite directional response to adding 10 grams of nitrogen in this system, which is a really big boost to productivity. So these effects of community responses, again, are really key to understanding ecosystem function, and they can be really on par with a lot of other environmental drivers. So of course, there's limitations to species removals as well. Thinking again about the applicability to nature, we know that dominant species are less likely to be lost with global change, although with many types of land use change, they could potentially be lost going forward. They also have a really high initial setup effort, and doing these manipulations opens up a lot of space. And so it's changing environmental conditions along with changing the species assemblage in our plots. And again, they're very difficult to scale up. So we can do this at small spatial scales, but removing these dominant species or any species across large spatial scales can be really difficult to set up and maintain. So overall, we know that communities are changing around the world in response to environmental conditions, either observationally or experimentally imposed global change drivers. We also know that community change has consequences for ecosystem function. So most of the studies we've looked at have thought about primary productivity in our systems, but we can think about this in the context of a wide variety of other ecosystem functions as well. And finally, we can think that experiments that are manipulating community composition can help us to uncover the mechanisms of community change and links of community changes to the functions that we care about in our ecosystems. So I'm looking forward to discussing this more with everybody today during the workshop, and thank you very much for listening.